I'm Pichen. I'm Sunita.
And I'm Kennedy. We are from Group 6.
So today we will do an experiment on determination of bacteria present in the environment and identification of bacteria with spore and moles.
First of all, take 3 petri dishes of nutrient agar and label each of them as 1, 2, and 3. For petri dish number 1, draw a line under the dish to give 2 sections. On one section of the dish, place your finger slowly on the agar. After that, wash your finger thoroughly using soap and running water and dry. By using the same finger, slowly place it on the agar of the other section. Next is take a cotton swab and rub at any parts of the body. Then rub slowly the cotton bud on the petri dish number 2. After that, take another cotton swab and rub at any places that you think contain bacteria, which is our group use a smartphone. Then rub slowly the cotton swab on the petri dish number 3. For the remaining 2 dishes, label them as A and B. After that, expose petri dish A to the air for 1 hour, while petri dish B will act as a control. Then, close the lid of all petri dishes and incubate all of the petri dishes at 37 degrees Celsius for 24 to 48 hours. After 48 hours, we observe and record all the growth in the petri dishes. After that, we do gram staining on the petri dishes number 1, 2, 3, A in loop. Add 1 drop of water to the slide. Then bring the inoculating loop over the flame to sterilize it. Next, take 1 to 2 colonies of bacteria on the surface of agar and mix them well using a sterile inoculating loop. Smear the mixture to the size of 5 cent coin in order to get a thin layer of bacterial smear. Then bring the inoculating loop over the flame again to sterilize it. Air dry the slide. To heat fix the slide, pass through the slide several times through a flame. The slide should not become too hot to touch, with the bacterial smear facing up. Cover the bacterial smear with a few drops of crystal violet, which is a primary stain, for 1 minute. Then gently wash off the slide with a slow running water. Add a few drops of iodine on the bacteria smear for 1 minute and wash with water. After that, decolorize with a few drops of alcohol. This is the tricky step. Stop decolorizing with alcohol as soon as the purple color has stopped bleaching off the slide. Then wash the slide with a running water. Cover the bacteria smear with safranin for half to 2 minutes. Then wash the slide with the running water again. After that, blot the slide with towel tissue and observe the slide with the microscope.
To identify bacteria with spore. First, using a sterile inoculating loop by flaming it and let it cool, and then add a drop of sterile water on the glass slide. And then we sterile the wire loop again. Then we take 1 or 2 colonies of bacteria on the surface of agar and mix them well using the sterile inoculating loop. We sterile the wire loop again. We close the specimen. We air dry the slide, and to fix the slide, pass through the slide several times through the flame with the bacteria smear facing up. Next, we carry out the crystal violet stain by covering the bacteria smear with a few drops of crystal violet for 1 minute. After 1 minute, we gently wash off the slide with a slow running water. Then blot the slide with towel tissue. And then add a drop of oil on the smear and cover it. We can further carry on with the observation on the microscope. to observe for the morphology, color, and shape of the bacteria. The results are recorded in the table. Next, we are going to carry out malachite green staining. We sterile the wire loop and then add 1 drop of sterile water on the glass slide. Then we sterile the wire loop again. We take the 1 or 2 colony using the wire loop and smear on the glass slide. Then we flame the wire loop again to surround it. And we air dry the glass slide above the fire. Then we heat the beaker containing water until boiling. And we wet the smear with a few drops of malachite green and place the slide on beaker with the boiling water for 10 minutes. Do not allow the slide to dry. When it almost dry, we can add more malachite green if needed. After 10 minutes, we continue to wash the slide slowly under running water until the stain do not come out anymore. Next, we restaining with safranin for 2 minutes. After 2 minutes, we wash slowly under running water. Then we blot dry with the towel. And the step is repeated for the observation under microscope. To identify bacteria and mold using wet mount. First, we sterile the wire loop under the flame. Add a drop of water on the slide. Surround the wire loop again. Take a little bit of mold from the colony of bread and put on the slide. Remember, do not smear the colony to make sure we don't break the structure of the bacteria. And cover slowly with the slide cover. Observe under microscope using 45 times objective length and draw for the observation. Thank you.